Hi, physiology students, and welcome to Chapter 16, which is on blood. This PowerPoint is really lengthy, as you can see, but again, this textbook is an anatomy and physiology textbook. So a lot of about blood you should have gotten from anatomy. So some of these slides will be a review from anatomy, and then we'll focus. You'll obviously be responsible for all the information still, but I'll try to focus on some more of the physiological concepts about blood. So some anatomy review, and I'll go over these slides rather quickly. The functions of blood, as you remember, work in the transport of molecules, regulating fluid levels and other ion concentration levels, and also help to protect. So they will transport all your white blood cells that help in the immune response. So transport includes delivering O2, transporting wastes, transporting hormones. So this is the transport function of blood. The regulation of blood helps to maintain temperature regulation, maintaining pH using buffers, and maintaining fluid volume within the circulatory system. Protection includes preventing blood loss by initiating clotting formations, preventing infection by carrying antibodies, proteins, and white blood cells. So the composition of blood, it's the only fluid tissue in the body. The matrix is non-living plasma, and then cells are the living blood cells called the formed elements. So to make up blood, we get plasma and formed elements. Plasma is non-living and formed elements are the living cells. So within the formed elements, we have erythrocytes, leukocytes, and platelets, which are the three types of cells: red blood cells, white blood cells, and platelets. If we spin blood down, we'll get three layers. Erythrocytes will be on the bottom. So let's say we take a blood sample from you to check your white blood cell concentration. We centrifuge it down, which is an extremely fast spinning, and then the blood will divide itself into these three layers. The erythrocytes will be on the bottom. The buffy coat includes the leukocytes and your platelets. The leukocytes are your white blood cells, and then the plasma will float to the top. And this is kind of the separation of those. The hematocrit is the percent of blood volume that is only red blood cells, and it changes a little bit on your gender and age. The erythrocytes make up about 45% of whole blood. The white blood cells and platelets make up less than 1%, and the plasma always floats up to the top, and that makes up about 55%. Blood is very sticky, opaque, fluid. The color varies with oxygen content. So a high oxygen level shows a scarlet red color, and lower oxygen levels show a darker red color. So there's a little bit of a difference of color between high oxygen levels, which will be in the arteries directly leaving from the heart, and lower oxygen levels, which will be in the veins, bringing blood back to the heart. The pH of blood is right around 7.4, and anything above or below this pH range will kick into negative feedback mechanisms, which will try to get the blood back into normal pH range. Your blood makes up about 8% of your body weight, and the volume of blood differs slightly if you're a male or a female. Blood plasma is straw colored. It's made up mostly of water. There's over 100 dissolved solutes in plasma. Plasma proteins are the most abundant solutes, and the albumin is the most abundant plasma protein. And albumin functions as a carrier of other molecules, as a blood buffer, and contributes to the plasma osmotic pressure. So here's a composition of plasma. You can see it's mostly made up of water, solutes, and the big protein that makes up plasma is albumin. So the formed elements then, we have red blood cells, white blood cells, and platelets. Only white blood cells are complete cells because your red blood cells have no nuclei or other organelles. And that's because the red blood cells are formed to try to pack in as much oxygen as possible. So there's no room for any sort of nucleus or other organelles. Most formed elements survive in the bloodstream only for a few days, and most blood cells originate in your bone marrow and will not divide. So here's a look at a sample of blood. You can see the erythrocytes without the nucleus. The leukocytes are the main formed cell, and then the platelets are a lot smaller, and the platelets help in blood clotting. And this is a photomicrograph under a microscope of what we would see a sample of blood. We're going to you are responsible for knowing the different types of leukocytes. Those are the cells with the kind of the darker colored centers. The red blood cells are kind of the donut shaped ones. And then I don't think you can just hardly see platelets in this sample as well. So here we'll talk a little bit about erythrocytes. They play a crucial role in oxygen and carbon dioxide transport. And that's because of their structural characteristic and shape. They're filled with the protein called hemoglobin that is for the gas transport of oxygen and carbon dioxide. You should be familiar with what a red blood cell looks like. It's biconcave. It contains the plasma membrane protein spectrin to help provide flexibility and change shape. The superb example of complementary of structure and function. This just describes that the red blood cell's structure is directly attached to the function of it. Three features make the red blood cell very efficient for gas transport. The biconcave shape offers the huge surface area. Hemoglobin makes up 97% of the cell volume, and hemoglobin is the protein that transports oxygen. Red blood cells have no mitochondria, so their ATP production is anaerobic. What that means is that they're not consuming the oxygen they transport, which is important. So all the oxygen that red blood cells are transporting is going directly to the tissues. So here's a structure of erythrocytes, the red blood cells. The biconcave disc shape means it's just indented on the top and bottom, kind of like a donut. Red blood cells are dedicated to respiratory gas transport. Hemoglobin binds reversibly with oxygen, meaning oxygen can bind to hemoglobin and then be offloaded. Here are the normal values for hemoglobin in males and females. Hemoglobin consists of a red heme pigment bound to the protein globin, and globin is what is composed of 4 polypeptide chains, 2 alpha and 2 beta. A heme pigment is bonded to each globin chain, which gives the blood a reddish color, and each heme central iron atom will bind 1 oxygen. So each hemoglobin, because it's made up of 4 chains, can bind up to 4 oxygens. So here's a look at the structure of hemoglobin made up of the 2 alpha globin chains on the bottom, 2 beta globin chains on the top, and their central heme group with the iron atom binds to 1 oxygen. So 1 hemoglobin molecule can bind 4 oxygens. And here's a look at the structure of hemoglobin, the chemical formula showing the 4 chains and then the iron-containing heme pigment. So each hemoglobin molecule can transport 4 oxygens. So this means that each red blood cell contains 250 million hemoglobin molecules. So 250 hemoglobin molecules, each transporting 4 oxygens, means that 1 red blood cell can contain up to 1 trillion oxygen molecules. Oxygen loading in lungs produces oxyhemoglobin, which makes it a ruby red color. And oxygen unloading in tissues produces deoxyhemoglobin or reduced hemoglobin, which is a darker red. Carbon dioxide loading in tissues, 20% of carbon dioxide in the blood binds to hemoglobin as well to help in the transport of the waste of carbon dioxide back to the lungs. And that produces what we call carbaminohemoglobin. Hematopoiesis is the formation of all red blood cells. It occurs in the red bone marrow. Hematopoietic stem cells are stem cells that give rise to all formed cellular elements. And new blood cells enter the blood sinusoids, which are the kind of structures that will take the blood. Stages of erythropoiesis. Erythropoiesis is just the process of the formation of red blood cells that takes about 15 days. It occurs in the bone marrow. The stages of transformation are shown here. You don't need to know all the stages, but you should know the main hormone that initiates erythropoiesis. And this is how we get the formation of red blood cells. We create a stem cell, a committed cell that will then go on to form an erythrocyte in the different stages of that. And you can see here where the nucleus eventually gets removed to create the erythrocyte. Regulation and requirements of erythropoiesis. So too few red blood cells leads to tissue hypoxia, and too many increases the blood viscosity or the thickness. About 2 million red blood cells are made per second, and the balance between red blood cell production and destruction depends on hormonal controls and dietary requirements. Here is the big hormone that you should know. Erythropoietin is the hormone that stimulates the formation of red blood cells. There's always a small amount of it in the blood to maintain the normal red blood cell production rate. And the big thing you should know is that erythropoietin is released by your kidneys, some from the liver, but mostly by your kidneys in response to low oxygen levels. So erythropoietin will be released from your kidneys if oxygen levels get too low to create more red blood cells. Causes of hypoxia can be decreased red blood cell numbers due to hemorrhage or increased destruction. Iron deficiency would be an insufficient hemoglobin per red blood cell ratio, or reduced availability of oxygen, for example, at higher altitudes, or lung problems such as pneumonia. Too many erythrocytes or high oxygen levels in the blood inhibit erythropoietin production, so it causes erythrocytes to mature faster. Testosterone also enhances erythropoietin production, resulting in higher red blood cell counts, usually in males. So here's just kind of the homeostatic balance that we often show in regards to many systems in the body. This is showing normal blood oxygen levels. And if that balance becomes a little bit off, your kidneys, again, will release erythropoietin, which will stimulate red bone marrow production of red blood cells in the body. Some athletes will abuse artificial erythropoietin, so they use erythropoietin supplements to increase their hematocrit or red blood cell formation, which will increase stamina and performance. There are dangerous consequences to this. So for example, erythropoietin can increase the hematocrit from 45% to 65%, with dehydration concentrating the blood even more. So this can eventually lead to the blood becoming like sludge and can cause blood clotting, stroke, or even heart failure. Dietary requirements for erythropoietins: you need iron, amino acids, lipids. You get most of your iron from your diet. So free, and that's why without iron, people that can cause problems if there is not enough iron in the diet. Vitamin B12 and folic acid are also necessary for DNA synthesis for rapidly dividing cells like developing red blood cells. The lifespan of your red blood cells is about 100 to 120 days. They are anucleate. They don't have a nucleus, so they can't synthesize new proteins or grow or divide. So once they become fragile, hemoglobin will degenerate. They can get trapped in smaller circulatory channels like your spleen, and macrophages in your spleen will engulf and break down dying red blood cells. Red blood cell breakdown, so the heme, iron, and globin will be separated and basically recycled. So heme will be degraded to the yellow pigment bilirubin. The liver secretes bilirubin into your intestines, where it's degraded into urobilinogen, and that will be transformed and leave the body in feces. Globin will be metabolized into amino acids, which will be released into circulation. So this just shows you how the life cycle of your red blood cells will kind of be recycled and used. The breakdown products of it will be used as well. Most erythrocyte disorders are classified as either anemia or polycythemia. Anemia is abnormally low oxygen levels. Symptoms can be fatigue, pallor, dyspnea, and chills. And there's 3 groups based on the cause: either blood loss, not enough red blood cell production, or too many red blood cells being destroyed. Blood loss, we can get hemorrhagic anemia, which is rapid blood loss in a severe wound. Chronic anemia could be due to hemorrhoids or bleeding ulcers. Not enough red blood cells being produced could produce what we call iron deficiency anemia. Can be caused by hemorrhagic anemia, but also a low iron intake. So a treatment for this is iron supplements. Not enough red blood cells being produced can also cause what we call pernicious anemia, which is an autoimmune disease that destroys the stomach mucosa. Intrinsic factors needed to absorb B12, and B12 is needed to help red blood cells divide. So without B12, red blood cells will continue to enlarge but not divide. So the treatment is B12 injections or a nasal gel. Renal anemia is caused by a lack of erythropoietin, accompanied by renal disease. Your kidneys cannot produce enough of that hormone erythropoietin, so your red blood cells cannot be made, and the treatment is synthetic erythropoietin injections. Aplastic anemia is destruction or inhibition of red bone marrow, can be caused by drugs, chemicals, radiation, or viruses, and a treatment is usually short-term with transfusions and long-term with transplanted stem cells. Too many red blood cells destroyed also is a type of anemia. This is called hemolytic anemia, caused by some sort of disorder like thalassemus or sickle cell anemia, which is a genetic disorder of the red blood cell itself. And you can read through the too many red blood cells destroyed here. And sickle cell anemia, it's more prevalent in African Americans, usually due to malaria. Possible benefit is that people with sickle cell anemia do not contract malaria at all, but it does kill about 1 million per year. And sickle cell anemia is passed on genetically. And we'll talk about that when we get to genetics a little more, but that's sickle cell anemia. Treatment is usually gene therapy, stem cell treatment, acute crisis treated with transfusions, inhaling nitric oxide. So this is a look at a normal red blood cell and what sickle cell red blood cells look like. Polycythemia is an abnormal excess of red blood cells, increasing the blood viscosity, causing sluggish blood flow, bone marrow cancer leading to excess red blood cells. Secondary polycythemia can be caused by low oxygen levels or increased erythropoietin production. Blood doping is when athletes remove, store, or reinfuse red blood cells before an event to increase oxygen levels for stamina. Not recommended. So the leukocytes for this section, I'm going to let you guys read through the leukocytes section on your own. It goes through the 5 different types of leukocytes, which are red blood cells. They're the only formed element that is a complete cell with nuclei and organelles. The leukocytes usually increase in a normal response to infection. So you'll often, if you are sick, they might measure your white blood cells to see if your body's trying to fight off an infection. They can be granulocytes or agranulocytes, and there's a mnemonic to remember with decreasing abundance in blood, the 5 types of leukocytes. So I'm going to skip over this section. You guys are responsible for this information. I'm hoping you touched on this in anatomy. So review this from anatomy, the 5 types of leukocytes, knowing what they look like, which is the most prevalent, starting with the granulocytes and then going to the agranulocytes. So that is the leukocyte section. And I'm going to skip over that again. So thanks for waiting as I go through this slide. And I'm trying to see if there's anything you guys do not, one thing you do not need to know is kind of the production and lifespan of leukocytes, but you should know the 5 types of leukocytes. All right, leukocyte disorders. You should know leukopenia is an abnormally low white blood cell count. Leukemia is a cancerous condition involving overproduction of abnormal white blood cells. So that would be a leukemia, and there's just different types of leukemia. Infectious mononucleosis is a highly contagious viral disease having to do with your leukocytes as well. The platelets are the fragments of larger megakaryocytes, and their function is to form a temporary platelet plug that helps to seal breaks in blood vessels. Circulating platelets are kept inactive and mobile by nitric oxide. So you should know what platelets are and why they are important. In terms of platelet formation, it's regulated by a hormone called thrombopoietin. You don't need to know necessarily the formation of platelets, but you should know why platelets are important. They help to form blood clots. This is a good summary of the formed elements in the blood. And again, a lot of these tables, knowing the information in these tables is really helpful to learning all the questions that I might ask you on an exam. Hemostasis is the fast series of reactions for stoppage of bleeding. This requires clotting factors and substances released by platelets and injured tissues. There's 3 steps involved in hemostasis: the vascular spasm, the platelet plug formation, and then the coagulation, blood clotting. You don't need to know specifics of each step, but you should know what is involved in each step because this is very important in forming blood clots. So vascular spasm is step one. It's triggered by some sort of injury. It can significantly reduce blood flow. So it kind of is just when a vessel responds to injury by vasoconstricting. So that's a vascular spasm. The platelet plug formation are when the platelets stick to collagen fibers that are exposed. So they try to stick together to stabilize a platelet collagen adhesion. More platelet plug formation. We have platelets swelling, becoming spiked and sticky, releasing chemicals that produce a positive feedback mechanism as more platelets stick together, which cause more platelets to stick and release more chemicals. Platelet plugs are good for small vessel tears, but larger breaks in vessels need an additional step called coagulation. Coagulation is actually the blood clotting that reinforces the platelet plug with fibrin threads. There's a series of reactions that use clotting factors. Coagulation itself occurs in 3 phases, which I won't have you guys know, but you should know these events of hemostasis, the vascular spasm, platelet plug formation, and then coagulation. This is just a zoomed in look at each: the vascular spasm, the constriction of the blood vessel, the platelet plug formation, and then the coagulation step. I'm going to skip over the phases of coagulation. And you guys don't need to know those, but there's 3 stages itself of coagulation that you guys don't need to know, but the coagulation is just all the anticoagulants, the factors that come together to help to clot blood. Okay, this is a cool picture, the scanning electron micrograph of the erythrocytes trapped in the fibrin mesh that's trying to coagulate or form a clot in the blood. And excuse me, an anticoagulant would do the opposite effects to try to decrease that. So here's a look at all the blood clotting factors in the blood, their function, where they come from. And again, clotting your blood is good in a sense that it helps to respond to any sort of injury in a blood vessel. It'll try to clot the blood so that you don't lose the blood through a damaged tissue. The clot must be stabilized and removed when the damage has been repaired. So clot retraction is also a process. And this takes you through the steps of clot retraction. So once we create the clot and coagulate the blood together, we need to get rid of the clot eventually. Platelet-derived growth factor is released by your platelets. And then we have the fibrinolysis, which is a process whereby clots are removed after the repair is completed. Factors limiting normal clot growth, and factors preventing undesirable clotting. There's 2 major types of disorders. An undesirable clot formation is thromboembolic disorder. So whenever you see the word thrombo or thrombus, that has to do with a blood clot. And a bleeding disorder is an abnormality that prevents normal clot formation. And I'll let you guys read through those disorders on your own, understanding what a thrombus is. It's a clot that develops and persists in an unbroken blood vessel. An embolus is a thrombus freely floating, and an embolism is an embolus obstructing a blood vessel. So these 3 terms you should be familiar with. Anticoagulant drugs are used to prevent an undesirable clotting. So we have aspirin, heparin, and warfarin. A bleeding disorder's thrombocytopenia is a deficient number of circulating platelets. And this is a look at petechiae, which appears as a result of spontaneous widespread hemorrhaging. So you get this hemorrhaging within the blood, and you can see it sometimes as red dots persisting in the feet. Other bleeding disorders can be from impaired liver function. Hemophilia is a hereditary bleeding disorder. DIC involves both widespread clotting and severe bleeding. Cardiovascular system minimizes effects of blood loss by reducing volume of affected blood vessels and stepping up production of red blood cells. So here we're going to get into blood transfusions a little bit. Restoring blood volume. Volume must be replaced immediately after shock. normal saline or some sort of electrolyte solution can be given. Whole blood transfusions are used only when blood loss is rapid and substantial. Infusions of packed red blood cells will be preferred to restore oxygen carrying capacity. And here is where we're going to get into some blood typing and the human blood groups that we'll talk about now. Okay, so blood typing groups, you should know very well about the different blood typing groups. Red blood cells have different antigens, and an antigen is anything perceived as foreign that can generate an immune response. So your red blood cell antigens are referred to as agglutinogens because they promote agglutination. And a mismatched transfused blood is perceived as a foreign and may be agglutinated and destroyed, and this can be a potentially fatal reaction. Humans have at least 30 naturally occurring red blood cell antigens, but the main ones that we will talk about are the antigens of the ABO and RH blood groups. They cause the most vigorous transfusion reactions if mismatched, and therefore they are the major groups typed. So ABO blood groups, so your blood type is based on the presence or absence of either A or B agglutinogen on the surface of your red blood cells. So if you're type A blood, you have the A agglutinogen. If you're type B, you have the B agglutinogen. If you're type AB blood, you have both. And if you're type O, you have neither. Blood can contain preformed anti-A or anti-B antibodies, which we'll talk about as well. And they just act as against transfused red blood cells with ABO antigens that are not matching. Anti-A and anti-B antibodies will be formed in the blood at about 2 months of age, reaching adult levels by 8 to 10. So here we have ABO blood groups. And again, your blood group is named for what type of agglutinogens are present. So your red blood cell antigens, if you're AB, you have both A and B antigens on the surface of your red blood cells, which are seen here. B's are the green circle antigens and A's are the pointed antigens. If you're type B blood, you just have the type B antigen on the surface, but in your plasma, you'll have anti-A antibodies, meaning if you're type B blood, you have the B antigen on the surface of your red blood cells, but circulating in your plasma, you have anti-A antibodies to fight off against the A antigen if you have a mismatched. Here's the blood that it can be received from, and just the frequency of the US population in what type of blood you have. Blood group A has the A antigen and has anti-B antibodies circulating. And if you're type O blood, you don't have either antigen on the surface of your red blood cell, but you do have both types of antibodies circulating in the plasma. So type O blood is also often known as the universal donor because it doesn't have any antigens on the surface of its red blood cells. Its red blood cells won't be interacted or be kind of fought off by any recipient blood. So if you're type O blood, you can donate to anyone. And if you're type AB blood, you're the universal recipient because you have both antigens on the surface of your red blood cells and you don't have any antibodies circulating. You won't have any sort of interaction, so you can receive any type of blood. RH blood groups then give you the positive or negative that you often hear associated with your blood type. So RH positive indicates the presence of the D antigen, and anti-RH antibodies are not spontaneously formed unless you're exposed to it. For example, if an anti-RH antibody forms, only if an RH negative individual receives RH positive blood. So you will only form antibodies against this RH antigen if you're exposed to it. This could happen in the fetus. So if an RH negative mom is carrying an RH positive fetus, the mom might actually start to create antibodies against the RH positive antigen on the fetus's blood. This won't be a problem for the first pregnancy, but subsequent pregnancies could be a problem because now the mom has these antibodies against the RH positive factor that could harm the fetal blood. Hemolytic disease of the newborn is also called erythroblastosis. This occurs when an RH negative mom has an RH positive fetus. So during the first pregnancy, the Rh negative mom is exposed to the Rh positive blood. So the mom will begin to create antibodies against that Rh positive factor. The first baby is born healthy, but the mother during this pregnancy creates antibodies against that Rh positive. So if we have a second pregnancy, the mom's anti-Rh antibodies could cross the placenta and destroy future Rh positive blood of a second pregnancy. What can happen is the baby can be treated with pre-birth transfusions and exchange transfusions after birth. RhoGAM is also a serum containing anti-Rh, which can prevent the mother from being sensitized to those antibodies. A transfusion reaction occurs if mismatched blood is infused. The donor cells will attack or be attacked by the recipient's plasma agglutinins. They will cause an agglutination or a clumping, which can clog small vessels and also rupture and release hemoglobin into the bloodstream. This will result in diminished oxygen carrying capacity, decreased blood flow beyond the blocked vessel, and hemoglobin in your kidney tubules can lead to renal failure. The transfusion reaction, a symptom could be fever, chills, low blood pressure, rapid heartbeat, nausea, vomiting. The treatment is to prevent kidney damage with fluids and diuretics to wash out the hemoglobin, give type O blood, which is the universal donor. Type AB blood is the universal recipient. So you should have a good understanding of blood typing. Blood typing is when the donor blood is mixed with antibodies against the common agglutinogen. If agglutinogen is present, the clumping of red blood cells will occur because they're attacking each other. Blood is typed for the ABO and for the Rh factor in the same manner. Cross-matching is typing between a specific donor and specific recipient so that you can mix the recipient serum with donor red blood cells, mix the recipient with donor serum to make sure no agglutination would occur. So this is mixing blood types together and showing agglutination is the clumping that occurs. And the clumping that occurs just shows that the antibody has found the antigen and has clumped. So this is type AB blood containing both A and B antigens. And if it's mixed with anti-A and anti-B antibodies, it will clump because the antibodies are clumping with the antigens present. Type A blood containing the A antigen will clump if mixed with the anti-A antibody. So this kind of picture here is showing that this is a sample of type A blood because the anti-A antibody has found the type A antigen to clump with. Type B blood has the B antigen that'll clump with the anti-B antibody. And type O blood doesn't contain any antigens on the surface of the red blood cells, so no clumping will be seen when mixed with the different antibodies. Here's an examination of blood that can yield information on a person's health. A low hematocrit is seen in cases of anemia. Blood glucose tests check for diabetes. Leukocytosis can signal an infection. And a microscopic examination can also reveal any variations in the size or shape of red blood cells. Other diagnostic blood cell tests, differential white blood cell count looks at the relative proportion of each red blood cell. Prothrombin time and platelet counts assess hemostasis. CMP is the comprehensive medical panel, and complete blood count checks for formed elements, hematocrit, and hemoglobin. The lab on blood in your PhysioX will take you through some of these same things that we talked about. But that is the end of chapter 16 on blood. So thanks for listening, guys.